We are ready. Welcome. Welcome, Alfonso. Welcome to the audience. Day 2 of the Bertalanffy Lecture, and today it's really for the campus, for the scientists, and I'm pretty sure that there is scientists more experienced, but also starting scientists in the audience. And in today's presentation, Alfonso is going to be a bit more provocative than yesterday, I think, and well, let me properly introduce him. Alfonso Martinez Arias does not need an introduction because you're here even without me introducing him. He's a biologist. He studied biology, got a degree in biophysics in Chicago, and from Chicago went to Cambridge, and I think this is where we, I think, still mentally have him localized. He's not in Cambridge any longer. He's in Barcelona since 21. And I think that is a pretty sharp transition after many, many years. I think more years in Cambridge than I'm married, which means something, or not. But he's now in Barcelona, and he continues things on cells. So he's not obsessed with cells, but he's clearly stating that cells are the basis for everything. And if you want to, you can engage him into a debate whether it's the genome or the cell, but the cell will win. So you may want to start it or leave it for something later after a longer discussion in a different direction. So today, where is it? I need the title. He's going to tell us about stem cell models of mammalian embryogenesis. This is what he's working on since more than 10 years. In a sharp switch from Drosophila. And I think today the provocative part is the second sentence, hope or hype. And I'm pretty sure you will get both hopes and you will also see the hype. Alfonso, as soon as...
Well, talking about hope, there we
yes. are. No, there's always hope.
So thank you, Jochen. It's always a pleasure to be here in Heidelberg and to be at KOS. I think Jochen has been very kind to me over the years, inviting me to come to Heidelberg to talk to many generations of students. And I've always enjoyed coming here to talk to students and to share science and interest with Jochen and his colleagues. Many of them has become my friends. He said I was going to be more provocative. That is not true. Yesterday I intended to be provocative. Today I try to tell you about my science, and that's what I'm trying to do. And I don't think there is any provocative there, but we'll see. The lectures, these lectures that Jochen never explained to me why he called the Ludwig von Bertalanffy Lectures. Ludwig von Bertalanffy will not mean very much to any of you, but Ludwig von Bertalanffy is the first person that talks about the need for what we call today a systems theory of nature. And he was even broader than biology. And he thought that the way to understand nature in any of its parts was not to analyze its component parts, but to see how they come together. And as you will see today, sometimes in order to understand or in order to query nature, you don't need to query the parts. You have to go to the level that nature works. And I hope that as you will see today in the talk, that is what some of us are trying to do in developmental biology. The problem with that is that you walk into the unknown. And all we can do is highlight the questions that unknown produces and hope that you will help us or help yourself find the answers. And I hope that a lot of what I tell you will stimulate your imagination and will stimulate. Now, I had a problem with the title. And the problem is that the first part of the title, which is what Jochen could read, that's fine, but then I realized in preparing the lecture that I had talked about hype or hope. And that's something that maybe is in the back of my mind when I've been moving into this field and for many things. And so I will address what I mean by hype or hope. Let's start at the very beginning, and the very beginning is the creation. And I think it's quite fascinating how maybe because of this kind of myth over the years, we, as part of the human race, have been obsessed with building a human, maybe not in the way God did it, which he knows how he did it, but in different ways. And that has led over the years to a number of possibilities. On the left, you have a Jewish myth, which is the golem, which they build, and they can do things. In the middle, it was called Frankenstein. It's interesting that in both cases, there is a little girl next to it, and that's automata that I talked yesterday about. But there has been this great interest of how could we build a human being. As a scientist, there are interesting questions in that endeavor, but as human beings, there is no question that has been an obsession that runs through the history of humankind and through the myths. Over the last few years, we seem to have found a way of maybe doing this, and this is, for example, an example of last year. There you have, scientists create living entities in the lab that closely resemble human embryos. I mean, it's there you go. This is Dr. Polo in Australia with a lot of pictures of human blastocysts, very proudly saying, we built something without the sperm and eggs, and this is... The problem is that when we look carefully at those structures, they have nothing to do with the human blastocyst. They resemble it, but it's not closely, and you cannot go anywhere. Things got even more heady recently when you get these pictures, these headlines, a synthetic embryo made without sperm and egg. Those blastocysts, they were made also without sperm and egg. Could lead to infertility treatments, or even worse, could one day be used to create artificial human organs. I'll address those later, but that's the kind of a structure that you do, which I can assure you, it's a very messed up embryo. And you get all these headlines from Caltech, from the CNN, from the Daily Mail, from the Guardian, that you have made an embryo. But the question is, what is an embryo? And this is my view of what is an embryo. An embryo is an autonomous, integrated dynamical system. That goes to the heart of Bertalanffy. With multicellular scaling laws, spatiotemporal coordination of cell type specification into patterns. I was talking to my friend Lázaro about this. This is a question he's interested in. 3D axial organization. We have 3 axes. All of us share that, independently of how we look. And it's been honed in by millions of years of evolution. The way we are is not the product of some whim of someone building something in the lab. It's been honed in, is perfect. And we should think about the perfection. Now, if you think about it, those of you who are in biology, these days we get the impression that if we want to know how anything is made or how anything is constructed, all we have to do is find out the genes that they express, and that's what it is. So the question is the development of an organism just a mapping of genes space into cell space? Well, actually, I believe that it is all about mapping cells into space and time. And at the risk of repeating myself, but it's not important, I'd like to give you the best example that I know of how development is about mapping cells into space and time. And that is the process of gastrulation, and is this wonderful movie of the frog embryo during gastrulation. And you can see there that you have a mass of cells, and this mass of cells sort of rearranges itself in a manner that is very precise. It happens in the same way in every embryo. There you have the nervous system appearing. And then at the end, you get exactly what you are getting there. You get the embryo at the top, and below is a sagittal section so that you can see the organization of the different germ layers. The embryo at the top, it's a scheme of a frog embryo, and here you have now, that's the frog embryo, and below what you can see now, that's a human embryo. That's actually a human embryo. You can see the similarities. This is quite important to me. This is an embryo of about, I would say, 6 weeks, and you can see that it's very, very similar in the structure and organization to a human. A human embryo develops from a blastocyst that's sorry about this. That sort of summarized there the process of development of a human embryo. On the side is what happens, what you would have after a week of development is on your left, and the cells in green, that's what it's, the trophectoderm is around, that will give rise to the placenta. The cells in green will give rise to the embryo, and after another 7 days in which that group of cells implants in the uterus, gastrulation ensues, which is the second figure on the left. And then it continues to grow. In about a week, you can start seeing how that flat circle of cells has been transformed into something that you recognize as an organism with the head at one end and the tail in another. And that process, which is gastrulation in a human, lasts about a week. How do we know about that? Where this is part of a collection of human embryos that was gathered over many years in the Carnegie Institution in Washington. And we have a pretty good continuum of the development of the embryo. Why should we worry about these very early stages of development? Well, for a very simple reason that is sort of indicated here, and it is that it is during these early stages that most of the problems that happen during a pregnancy. So in fact, only about half of the fertilized eggs make it to gastrulation, and a lot of them are lost after gastrulation. If you go through implantation and gastrulation, okay, you're going to make it, and usually it's only about a third of the eggs that have been fertilized that make it to the end, only a third. And that third, many of them are going to have a lot of abnormalities that have been born during the process of gastrulation, and that usually, particularly heart conditions, are very, very common in a spinal cord organization. They are the most common pathologies that you can get. So it would be very, very important and useful if we could get an access to the mechanisms of implantation and gastrulation, but that is impossible because we cannot do experiments with humans, and we cannot also, it's not so easy. If some of you are thinking, well, we could use with primates that are very similar to us, that's even more complicated. You might not believe it, and that's because to develop experimental protocols and to keep many monkeys in a lab, it just costs a lot of money and also has ethical constraints that we should think about. So for many years, we have been using the mouse as a proxy for humans, and that's the... So how does gastrulation happen? See, the object of a lot of this is to try to understand gastrulation. There is a lot of people trying to understand the early stages and implantation. I'm not going to address that today. Maybe I'll touch upon it later. But I'm just interested in gastrulation because it's the most important moment in which what is a mass of equivalent cells get transformed into something that it's the blueprint, the sort of layout of the ground plan of the organism, in this case, the human or the mouse. So gastrulation in the mouse, it happens, you can see there a movie. It was filmed recently by Kate McDowell, now in Cambridge, in the Laboratory of Molecular Biology. That's one of her movies. But you can see that the first thing that happens in the mouse is this expansion of cells. There is a very, very important difference between early mammalian embryos and the embryos of any other species. And it is that you remember that frog embryo that I showed you, it was a ball containing thousands of cells. Turns out that the mammalian embryo, whether it is a mouse or it is a human, as it's going to gastrulation, in contrast with the thousands of cells that have the Xenopus embryo, a mammalian embryo, human or mouse, has about 300 cells. So the first thing that it has to do is expand those 300 cells into thousands, and it so happens that it does it through the process of gastrulation. At the same time, it's a remarkable process of self-organization, because what you get at the same time that the cells are expanding, the germ layers are being defined, and they are being organized in a space. Most important in gastrulation is getting a coordinate system. All this is happening simultaneously in the mouse. And then you can see indeed that according to that coordinate system, in this case is the cells that will give rise to the heart. You see them getting there, and after a while, you can see that those are nice embryos with the heart highlighted in green and yellow. And at the end, you have exactly at that stage. Usually this takes about a week in the mouse, as opposed to the 3 weeks that takes in humans. And you can see there that we have the body plan of an embryo with the tissues recognizable, the spinal cord in the middle, the cells that are going to give rise laterally. It's called the paraxial mesoderm on the side, an anterior head, and the tail at the other. But now, the most important element to me of this process of gastrulation is the generation of that coordinate system that is going to allocate the different primordia relative to each other and in the general position of the embryo. And that we don't know where that coordinate system comes from, but we do know how the embryo builds it. And the way the embryo builds it is using two cell populations that are established very early in development and that are not going to contribute to the embryo. They are derived from the zygote, but these cells are not going to contribute to the embryo. One is the, there was yesterday a green sticker, but anyway, I don't think it's that long. So the cells that you can see there, the cells at the top of the embryo, they are called the trophectoderm, and they're going to give rise to the placenta. And the cells around the embryo, you're looking at those cells that I'm highlighting in the, it's not going to reach. The cells that are green, it's called the primitive endoderm. They're going to give rise to the yolk sac, which is going to keep the embryo fed until the placenta takes over, which which surprisingly, the placenta starts feeding the embryo halfway through embryogenesis, in the mouse and in ourselves also, in humans. And it is interactions between those two extraembryonic populations and the embryonic populations that set up the axis, as you can see there. The first thing that happens in the first idea, the first realization, the first observation that there is an axis is the expression of the gene called brachyury that you can see here. And that's in a mouse. And you can see brachyury there, that's an in situ hybridization, and the arrows are highlighting the expression of this gene, which highlights the beginning of gastrulation and what will be the end of the body of the organism. That's the gene called brachyury. Brachyury is absolutely conserved in all animals, as you can see there in the graph on the left, and it is in green, and in all organisms, from sponges to us. I mean, that's a Darth Vader kind of thing. I don't think we're going to go very far. But anyway, you can see there. I think that's. Anyway, that was a good idea. But so it's interesting that this axis must have arisen very early in evolution because all organisms utilize brachyury positioned in one end to determine that axis. And the minute you get anteroposterior axis, you start being it always in the posterior, and you have it here in other mammals, in sheep, in cows, and in rabbits. Brachyury is always determining the posterior part, and it's the first sign that there is an axis in the embryo. You can see that in this scheme. And the way that happens is, as indicated there, first transition, the embryo goes from a mass of cells that are not very well organized to an epithelium, as you can see in the middle. The cells surrounding that epithelium that are in green, they are called the primitive ectoderm. That will give rise to the yolk sac. And now, the system is radially symmetric. Now, somehow, because of a signal from the trophoectoderm to the bottom of the primitive endoderm, some cells are selected that are going to move. They're endowed with the capacity to move. The cylinder, because we are looking at the cylinder, is radially symmetric. These cells move to one side. It could be any. That side will become the anterior part. They form what is called the AVE, for anterior visceral endoderm. And the posterior part of the embryo will be formed completely at the opposite end. That's where gastrulation starts. That's where brachyury starts. That's what you see in those sections of in situ in the rest. And that's the way the embryo acquires the first axis, which is the anteroposterior axis. It is through interactions between the extraembryonic tissues, bear this in mind, and the embryonic tissues. The embryonic tissues are the cells that will give rise to all our body parts and all the body parts of the embryo. How do we know about this? We know about this because of genetics, because over the last 40, 50 years, genes and genetic analysis have guided our understanding of the development and organs and tissues. And we've done that through mutants. And you can see there some examples of mutants of one of my favorite animals, the fly, and also of, in fact, what you have there is a mutant for brachyury. And what happens in a brachyury mutant is that you don't define the posterior part and you don't define gastrulation, you don't define the axis of the organism. So it's mutants, and mutants is what happens, what are the consequences of removing one part of the system, one gene. And then you have to interpret in those mutants what the bit that you've removed does. And that's not an easy thing, but that's what we've been doing for the last 30 or 40 years. However, as Jochen has introduced, and it's a little bit of an obsession of mine, it is cells that actually link and interpret and transform the information that lies in the DNA into tissues and organs. Just remember, and when you go, you can think about it, a gene without a cell is nothing. And if you want a very clear and close example of that, just think about the SARS virus, which the way it does it is by hijacking the cells. Otherwise, it would be inert and it would be very harmless. But it is when it interacts with the cell. It is the cell that suffers and the cells that is subverted. The same with the bacteriophages. So how can we learn? For the last 40 or 50 years, we've been learning a lot about development from genes. How can we learn from cells? Well, this is where things have changed over the last few years, and what we have been developing is engineering approaches. That's what people would like to say. I like to say playing with cells, really. Not to imitate. Many people would like, we're back in this idea. Let's use cells, and I'll tell you with cells in a minute, to try to build a human being or to try to build an organ. But actually, I think the way many of us are using cells is to learn what cells can do when you ask them to do things. And the cells that have been very important in this the development of this new way of doing developmental biology, which I think has emerged over the last 10 years, is embryonic stem cells. Embryonic stem cells are a very mammalian feature. I don't think they exist I was talking to someone this morning. One thing is pluripotent cells. I mean, all embryos have pluripotent cells in the beginning. They can give rise to all organisms. The cells in the mouse are pluripotent. There are no stem cells, embryonic stem cells in the mouse. Embryonic stem cells is something that we've been able to create from the pluripotent cells of the blastocyst if we culture them in vitro. The difference is these cells keep their pluripotency going forever. And you can test that because you can put them back in an organism. You can put them back in a blastocyst in that case, and then they will contribute to the mouse. Okay? But the stem cells don't exist in the mouse. This state is very transient in the mouse. But some people have managed, with a little bit of alchemy, to maintain that potency in culture forever. I should say, for those of you who are interested, that that potency doesn't last there forever. We know that over time, even these cells age and these cells mutate, and that's quite important. Over the last few years, we have been learning to, we realized that in vitro, we can get these cells to go through the same, don't worry about the details, this is just to show you a little bit that we've done our homework. We can actually guide them through the same developmental stages that the embryo goes through, that they would go in the embryo, and even develop them into different kinds of tissues and organs, as you can see there. And we play this by changing the media. It's interesting to me that the cells, what they're using in culture, when they're using in the embryo to communicate with each other, is a set of signals that some of you probably have heard of, Wnt, FGF, Notch, nodal, all these things. And those are the signals that the cells use to communicate with each other. What we do in order to transit the cells through these stages to generate the different tissue types and organizations that we know, it's, I think it's here, sorry about this, it's exactly the same language that they use to communicate with each other in the embryo. However, turns out that if you think about it, we've done this in culture. This was very encouraging when we realized this, that we could in culture guide the cells to the different tissues, to the components of the different tissues, but of course that is not the three-dimensional structure that is an embryo, whether a human or a mouse. In fact, we can do that both in the human and in the mouse, this game of oh yeah, we can have this arrow, that's very good. Is the same as that we can do that in vitro, but they are not organized in the proportions, they are not organized. So what would happen if we could create something that looks like an embryo? And this is back to the golem, to the Frankenstein, if you wish. And over the last few years, I told you that the beginning of the embryo is when these three tissues that are set up very early after fertilization, the trophoectoderm, the embryo proper, and the extraembryonic endoderm, people were able to derive stem cells for each of these cell populations. So the question is very simple. If the embryo arises from the interactions of these three populations, what would happen if we take these three stem cells, these three kinds of stem cells, and we put them together? Will we get an embryo? So many people have tried that, but in particular, there are a few labs, but one of them is by Magdalena Zernicka-Getz, which claims to have obtained some of these structures that look very much like an embryo. The problem that I have with this is that they only occur about, it's a game, it's a lottery. They mix these cells together, and at a certain proportion, that's usually less than 10%, you get these structures that resemble more or less the embryos. We repeated the experiment, and more or less you get the same problem. It's a bit of a mess, but you can fish out of there structures that do resemble very much the situation in the embryo. This is called the epiblast. The reason why I don't like that is because if we have been using organisms for years in order to study development, whether genetic or cellular, is because the embryo does it 100% of the time. And that's how we can analyze, and we can pick up subtle variations in the differences and learn what those subtle variations mean. If we are looking for an experimental system, it's very good that we can get these things, which, by the way, don't go farther than that. They don't even initiate gastrulation, or if they initiate gastrulation, they do it badly, but certainly they do it at a low proportion. Some people seem to be more interested in creating the golem or the Frankenstein or the automata made up of cells, so they don't worry about increasing the frequency. What they go is like in the circus. You remember some of you, I don't know, these days it's all on television. For those of us who are old enough to have been taken to the circus, and now more difficult. We're going to increase the difficulty of the event because we've got it. So they decided, two labs, but particularly Magdalena's, to instead of mixing the two of them, we're going to mix the three now instead of because the other one, I may forget that they were only using these two, so they keep on increasing the difficulty. This is in the trapeze. Now we remove the net, and now we see what happens. And some of you might have read that about a few months ago, and that's where those headlines that I mentioned in the beginning was overwhelmed by these structures, which, you know, what you have on the left are embryos. Sorry, what you have on the left here on the lower part, these are embryos. And these are, all these three are some part of these. There were two groups. One is led by Magdalena Zernica Getz, and the other is by Jacob Hanna in Israel. And, you know, you don't have the trained eye. That's why they can fool you, and they can fool the journalists. I can tell you that these are very, very sick embryos. These are not, they don't go further than, this is about a third of development. They're very good. They're a very good improvement. They have things, but there is no heart, there is beating cells. I mean, that I've discussed a little bit. There's a whole cardiomyocytes they are beating. There is the progenitor of a brain, but there is no brain, and there is no heart. This is a very early embryo with the primordia. More importantly to me, and this was given, the numbers were given by the Jacob Hanna group, which is the experiment very carefully. You can see that the frequency at which these structures arise on the plate is 0.5% of the starting material. So it's very good. I mean, that's almost like getting a golem or getting, well, a very sick golem or a very malfunctioning Frankenstein. But the important thing is that, okay, you can get it. This is a proof of principle. But I don't think this is going to be very helpful to test drugs or to obtain organs. 0.5% of the starting cultures. You have a long way to go. I should confess that many years ago, as we turn into embryonic stem cells, we did try to do these kind of experiments, but we didn't go very far because we didn't persevere. We decided to try more difficult. Let's just now got the trapeze up and remove the net completely. And we decided to ask a question that should have never been given the answer that it gave us. Remember, what you've seen there is that if you reconstruct, if you put together the three cell types that usually will give an embryo, they do it very badly, very poorly, and you don't go very far. Now the question is simpler than that. What happens if we just take the embryonic stem cells and we ask them, what can you do on your own? I'm sparing you all the tedious lab work to try to see if it works, and because what we got is here. That was very surprising. You can see a whole bunch of structures. Those of you who work with frogs, they might resemble you some very exogastrally, if you wish, although it's not the same. This is a technical detail. But what you can see is that this is not 0.5%. This is not 10%. This is 80%. And those structures, as I will show you in a minute, tell us a lot about embryos. They don't look like embryos at first sight, but they tell us a lot more than I'm going to be able to tell you about today. In fact, this happened 10 years ago, and I've spent the last 10 years trying to see what's the meaning of that. For many years, I've been thinking that this was a waste of time, but I think actually they are telling us, as I say, more interesting things that I'm able to tell you in this seminar today, but I'll be happy to discuss with you later. What have we done? And what can we do? Well, we call these things gastruloids for reasons that you will see in a minute, and it's basically that the outcome represents the outcome of gastrulation. So these things seem to have undergone gastrulation in a very mysterious way. We just take ES cells, we put them in a well, in a micro well, as you will see in a minute. We have to put very precise numbers of ES cells, and being alchemists of the 21st century, we have to do a little bit of magic. Add this Tinkerbell powder, which is called Wnt signaling, and then something happens, and you can see a little bit of what happens here. The cells come together. It's a round bottom plate. They come together, and then once aggregated, they start doing quite surprising things that surprise us. So that in the middle is using a cell line that has a gene for that SOC17, no, that's the brachyuria GFP. So all of a sudden, you can see brachyuria appears at one end of the aggregate. And if we wait a little bit longer, what you see is that those cells that started at 300, about the same number that they would be in the embryo, they start growing. You will see anteriorly that there are cells leaving the anterior part, and that's the neural crest, which is coming. And this continues to elongate and elongate and elongate. I should say that for a long time I thought that this was going to be voyeurism, and I didn't think. I was very surprised of what happened afterwards and what is happening. Particularly, I'm very grateful to the interest of Denis Duboul in this system, because it really, he had resources that we didn't have and helped us delve into that. So that's what of these structures look like if you start looking at the patterns of gene expression. There is a couple of things that is interesting. That is the progenitor of the heart. This is brachyuria at the end, and this is the beginning of a spinal cord. So that's basically the whole structure of the animal except for the brain. These things don't have a brain. How do we know that this is the whole structure of the animal? Before showing you that, I want to point out here that they develop a coordinate system exactly that of the embryo. From those 300 cells, no extraembryonic tissues here. So the cells have an ability to self-organize in a very precise and reproducible manner, the three axes. In fact, they even have a midline, and they can locate structures on the right or on the left. They do it randomly, which interestingly is what happens if you take an embryo out of certain membranes that it has and let it do the left-right asymmetry outside of the membranes that cover it in the embryo. And when we look at genes, we can see that the genes are organized along the anteroposterior axis, the dorsoventral axis, and also there is a midline. And when we look at the development of the cell types in these structures that we call gastruloids for the reason that they are the outcome of the process of gastrulation, you can see, those are details, you don't need to focus on the numbers. You can see that as you develop, the cells express more and more mature genes. So these things are being able to recapitulate without any external, without extraembryonic tissues, organize itself, and once they're organized, they go and run away. There is no brain. We know why. It has to do with the protocol. And as I tell people that get very surprised, the brain is not that important. I can tell you, I'm alive, and my brain is not functioning very well lately. Now, the most surprising thing, and this is what took the need, how do you define an organism? An organism, there is these Hox genes that many of you have heard about. They're a hallmark of animal development. We have 4 sets of them. They are organized in the chromosome in a manner that correlates with the expression along the anteroposterior axis of the organism, and also on the time. The most ones on your left are expressed first, and then they sweep. And you can see here, over time, I remember the day that Denis called me to show me this picture, you can see that as the development of the gastruloid progresses, this is the A cluster, this is the A cluster here, you can see that over time, these genes are expressed more and more. And that's what's happening as this thing is elongating. These genes are being unpacked, and they are being expressed from one end to another. And when we look at the expression in the gastruloids, as we call it, you can see that they have this nested pattern that is exactly the same as in the embryo. And Denis has just published a wonderful preprint, for those of you who are interested, using the gastruloids to unravel the mechanism whereby the expression propagates from one end to another. So this was the realization that this is telling us something quite intriguing and quite interesting about embryos, because, and bear that in mind, of course, they don't look quite like embryos, but they seem to have all the gene expression organization that an embryo has, without any of the morphology. So I think this is one of the things that I like after many years of thinking about it. I mean, if you try to make an embryo, you build the embryo, and then what have you learned? You are back where you were at the beginning. What is surprising is that cells that should not make an embryo on their own, make this structure that is so much as the abstraction of an embryo, but it's lacking something else, and I'll come back to that in a minute. A lot of these are structures that are posterior. What happens anteriorly? Well, anteriorly, when we look at the patterns of gene expression or the organization in the anterior part, we have a heart. I think you can see it there beating. This is a heart primordium. It's not a heart. And we know that because it has all the genes expressed. They have calcium waves, as the heart should be. And they also have placodalectodem, they have cranial mesodem, and they have neural crest. So these things are very organized. More importantly, I'm very worried about this. Oh, God. My slides have disappeared. Oh, no, they haven't disappeared. Oh, now it appears. Sorry. And all of a sudden, it had gone blank in my screen. So as I told you, one of the reasons, and you saw that yesterday, why we like to use embryos is because they do everything in synchrony. They do everything themselves. It's just, and I think that what is important in a system is not that you get something that you fish out that happens at 1% of, you need a system which you can use experimentally, therefore you need a system that is as robust as embryos, and this is what these things are. And you can see there how robust they are. Once we get the conditions right, this happens. 90% of the time, in the hands of some people, 100% of the time. We oscillate between 80 and 90. You can see here, this is work with Pierre-François Laine. These are ones that have not been treated with anything. The alchemy has not acted. In ones that the alchemy has acted, you can see that they are turning on brachyury. You can do high throughput. You can measure. They are very precise. These things are very precise. It's not like every one is of a certain, no, they are all the same length. They all have the genes expressed in the same manner. They are very proportionate. They are doing exactly what an embryo would do. So this allows us to do experiments, and this is ones that we are doing now and that I'm very, very intrigued and very hopeful, because as you can see, we have to put a very precise number of cells. If we put few cells, nothing happens. If we put too many cells, they get very messed up. But within a range of cells that goes in about 60, 70 to 300, you do form these gastruloids. And what is more intriguing is that the small ones form small gastruloids and the large ones form large gastruloids, but everything is proportional. Just like it happens in all of us, that we are different sizes, but they're very equally proportioned. So we hope that this will give us an entry into this most important and intriguing question of developmental biology. Also, I should remind you that the primordium of every organ also starts growing when it has a certain size. So this could be a representation from a lot of these very deep questions of biology. And we are now doing experiments to try to understand what's happening at this boundary, what is gained, what is lost at this boundary, and how this proportionality happens and what it means for the embryo. We've managed to do, we can look at one gene at a time, but in a very good collaboration with Alexander van Oudenaarde at the Hubrecht Institute in Utrecht. He had a technique which he calls tomoseq, in which you can take an embryo or a structure of any species and you slice it, just like a sausage. And then every slice you subject it to RNA-seq, and therefore you can get now how the expression of the genome is organized about whichever axis of your organism is. And we were very surprised to see, I mean, this has been a constant source of surprises, how these gastruloids, you can see this is here, then you need a bioinformatician, of course, to organize these things in space, that you can see that the genes are organized from anterior to posterior in a very pattern that we compare this to an embryo, except for the brain, is very, very good. So these genes are capable of being organized very much like we have what we would call a genetic blueprint of the organism in these gastruloids. What we noticed when we look at this, which was intriguing, is that there was a very strong signature of the process of somatogenesis, which, as you know, is the process that generates the somites. The somites are little epithelial balls that appear progressively during development that give rise to the cage, to your rib cage, to the spinal, to elements of the vertebrae, and to the muscles of the thorax from the neck down. And you can see here, this is from the, we could see this in the tomoseq, but we could also see it in the tomoseq and in the single cell. That's the progenitors of the muscles, of the somites. This is genes that are expressed in somites. This is very intriguing because in the organism, this gene, which is called RIPLY2, demarcates a boundary between the presomitic mesoderm, which has not undergone morphogenesis, and the somites, and I will show you that in a minute. When we compare these patterns of gene expression organized from posterior to anterior in the gastruloid with the data set that Olivier Pourquier had generated from the embryos that is similar, this is two gastruloids, this is an embryo, and you can see that we get a very, very good match. But when we look, this is somatogenesis going on. Here you have this presomitic mesoderm, which both express these genes like TBX6 and HES7 and delta-1. Now, this is the somites, which express ONCS 4.1, TBX8, PAS-A3, MUX2. You can see these genes are expressed here. This gene would be expressed at this boundary. And you can see that that's what happens. The layout of these patterns of gene expression is perfect in the gastruloid, but if you look at the gastruloids, there is no somites. There is no boundary between these two tissues. There is nothing. Emphasizing that these things seem to be missing some input, that although the cells are ready to generate morphology, they are not generating morphology. This is a very good example of a question that came out yesterday many times, and I'm sure it will come up today, that you can have all the genes expressed there, but the genes are not just on themselves determining the morphology. However, we did an experiment. I mean, there is two elements in the field of organoids today. I call it the Tinkerbell powder. There's two elements that form this magic potion, which is Wnt signaling and matrigel. Matrigel is a glue that is secreted by a cancerous cell line, and we all use these two things without really ever wondering which, so Tinkerbell powder. And when we put Tinkerbell powder into the gastruloids, you can see This is one of the first experiments. This is a gene that should be expressed only in the somites, and you can see that when we put it in matrigel, it starts segmenting, and we get some structures that are somite-like. But this was taken by students, Susan van den Brink, and I will show you something else in a minute, and took it further. And you can see that the first thing that happens, you get this beautiful segmentation. Just by putting there the thing in matrigel. It's the same genes, everything is the same, but now they're interacting with this matrix, which has mechanical and chemical components, and you get this thing now to segment, the cells to organize in a different manner. And Jesse Vibliet in Berlin and now in Dresden, he managed to even take this one better and generate these structures, which he likes to call trunk-like structures, in which you can see he gets a spinal cord, the presomitic mesoderm, and he gets very, very good somites. And this requires the interaction with the matrigel. What this says is that there is two elements in the system running in parallel. One is the genes, and the other is the cells. And we can separate them. That's what the gastruloids are doing, and I'll show you in a minute. The genes are creating gene regulatory networks using signaling molecules and transcription factors. The cells are using mechanics, and are using the environment, and are using numbers to create shape. What the gastruloids are doing is separating the two, but during development, they both come together. And we can see that in the process of that's the somites, and here for a moment you can see, because the slide is going to change. What I've just shown you is indeed that the cells, and that we can get now, we can get all these different things. We can get hearts, we can get even hindbrain and midbrain, we can get spinal cords, we can get somites, we can get guts, it's imperfect. Morphologically, it's imperfect. The important thing is the detail. But the fact that these things, out of 300 cells over the course of 5 days, they can generate this if you wish, abstract embryo, it's very remarkable in many ways. So what have we learned from all this over the last few years? There is a menagerie. This is over the last few years, following the kind of structures that I mentioned Magdalena is getting, and one can trace phylogenetic trees. But what are we learning? This is the thing that I'd like you to share with you. What are these structures telling us? Well, that these cells have the potential of self-organized axial organization, and that you might ask, what are then the extraembryonic tissues doing? Well, they are biasing that potential. They are ensuring that the embryo is oriented with regard to the mother in the correct way. So it's taking this intrinsic self-organizing potential, and it's ensuring the embryo has to implant and has to interact with the mother. So that's the main function, I think, of these extraembryonic tissues, to bias what is an intrinsic self-organizing potential to allow it to interact with the mother as development proceeds. Now, we've learned that cell fate assignments are independent of morphogenesis. I have shown you that, and that's going to be useful and interesting. Cell fate assignments require time. They respond to programs. Morphogenetic processes are space and create patterns. So I like to think of gene regulatory networks as creating time and morphogenesis as creating a space. And its evolution has brought them together in order to generate the wealth and majesty of organisms that we know. I didn't have time to discuss this, but the importance of the starting conditions. The state of the cells at the beginning of the experiment is absolutely crucial. And you can tell, this is not an experiment that works one day 20% of the time, another doesn't work. If it works 80% of the time. If it doesn't work, it doesn't work. And we know that that depends on the starting conditions. And finally, I think the embryo is modular. I think that this is very, very important. Once we had solved and sorted the question of the mouse, we thought, well, this may be a chance to try to get into human development, because there are human embryonic stem cells and there are iPS cells, and we did show that the iPS cells from the mouse would also make gastruloids. But we thought, well, maybe this is an opportunity of trying to tackle this mystery of the early development of the human being. And you know, we tend to forget human memory has a very short time span, and it does surprise me that many people that jump into this field of the human ESLs or trying to study embryos, they don't know that we do know a great deal about human embryos and human embryology. That's Wilhelm His, who was a Swiss-born embryologist of the 19th century. I was going to say past century, but I don't belong to the 19th century. That is the 19th century, and he is the first person that decided to try to understand human development. And he spent a lot of his time, and again, happy to discuss all the details of these stories, which are very interesting. Afterwards, he engaged a number of doctors and midwives to give him aborted embryos, and then he managed to organize them in a developmental series, and he is the first person this is from one of his works from 1885, in which he, for the first time, starts telling you how the human embryos develop. This is the seed of a very important collection of human embryos, which is the Carnegie collection. Franklin Moll was one of his students. He came to Europe to study with Wilhelm His, and then he took to America this idea of collecting human embryos and using them to create a very large timeline of human development that has become known as the Human Carnegie Collection. And Rohan O'Reilly and Fabiola Muller are people that have used this collection to make a very good account of embryology. Embryology, remember, is the description of the changes, macroscopic changes that cells go as embryos become more complex and structures and organs and tissues develop. It is thanks to this collection that we have learned a little bit about human development, and this is how we know that, in a way, the first part of human development can be divided into three parts. The early development, which is the cleavage of the embryo until it forms the blastocyst. That period lasts about a week. CS stands for Carnegie stage. This is stages that are normal in the referral to human development. Then the embryo is going to implant, and it takes a week to implant. The human embryo is particularly lengthy in this period. We know very little about this time, but we know that it's very important, because that's the moment where, if it doesn't implant or implants badly, embryogenesis is not going to proceed. That lasts a week, and after 2 weeks, you get gastrulation. This is the post-implantation period. And gastrulation lasts for another week, and then there will be all the time until 8 weeks when the fetus, when we transit from the embryo to the fetus. And as I said, it is the embryos of the Carnegie collection that has allowed us to get a glimpse of how the embryos look during this process and infer similarities and differences with other species. In fact, there is a lot of literature on medical embryology, which people forget, and that some of us are using to understand what I'm going to tell you about. Big difference between humans and mouse. Mouse have been described to you as a cylinder. The human embryo is a flat disc. But I should tell you that this is not because the mouse is the rule and the embryo and the human is weird. It is because the mouse is weird. In case you don't know, it turns out that 70% of mammalian species on Earth are rodents. 20% are bats, and the top 10-20% is the rest: the lions, the cows, the horses, the pigs, and us. And all of us have a flat disc, including bats. So the weird ones are the rodents. But that's why gastrulation here, it happens as a flat disc. And the only insight that we have into human gastrulation is from the embryos of the Carnegie collection. But as I told you before, a very important thing about gastrulation is that a lot of the defects that embryos, particularly the ones that make it to term, have, we think that they are generated in gastrulation. So it would be very good to have a model for that early human development so that we can get some inkling. I don't know whether some cure, but certainly some inkling into the early stages of development. You could say, well, we have been using mouse for many years, and we have learned a great deal that I think is transferable. But it is very clear that the way a human embryo uses the genes is different from the way a mouse embryo uses the genes. And this is an example. There is a lot of human genetics around. And this is Win3, is a gene that initiates the protein encoded by Win3 initiates gastrulation in the mouse. This is just for you to know that without Win3, gastrulation is messed up. The embryo does not develop the coordinate system and does not develop the fate of the cells that will give rise to a normal organism. This is from a family. This is a homozygous Win3 mutation in humans. And what you can see in these unfortunate pictures, the pictures of this unfortunate fetus, what you can see is these embryos have an axis, an anteroposterior axis, a normal anteroposterior axis, but lack the limbs. So this is showing you that Win3 is working, is being used in different ways in humans and in mouse. And what this says is that we cannot extrapolate what we know about mouse to humans in a very easy way, particularly because in humans it's not only to know about the development of the human embryo, but it's also trying to understand what the mutations are doing. So we have to be careful, and we need to develop systems that will allow us to study this, not just to look at the normal embryos. Human developmental biology, as opposed to embryology, is initiated by the development of in vitro fertilization by these two people here. I should change that picture, and today I will do it, because it should be three people, and the third person is a woman, and it's John Purdy, who was a technician with Robert Edwards in Cambridge, and she was absolutely fundamental. So when I see this picture, I always say I should change it, and I should change it, because he was absolutely instrumental in the development of in vitro fertilization. And the reason why developmental biology starts with these two people here, the three people, it's because you can imagine that this is not just about looking standstills of embryos fixed in formalin and try to describe them. This is about getting fertilization in vitro, and what happens when you fertilize the embryo, that now the embryo divides and it forms the blastocyst. I mean, that must have been a very exciting and fantastic moment in which you can see, and that's what developmental biology is about. Now you can analyze this, and it surprises me that this is not taught as part of developmental biology, and it's thought that this is a technique when there is a fantastic piece of biology and of the history of biology. But as you know, in 78, Louis Brown was born, and this was crucial. I'll tell you some interesting numbers about this later. But what is important about this is that because they had managed to get the human fertilized eggs all the way to the blastocyst in vitro, and then they had to implant, and things worked well, they questioned whether maybe they could continue that, and maybe they could get a human being in a dish one day. So they got very worried about this, and they had to wrestle with a number of interesting questions, from what is an embryo, what is an individual, and a committee under the leadership of Mary Warnock, who was a philosopher, an English philosopher. They convened the so-called Warnock Committee, which decided on several important issues concerning the development of human embryos in vitro. And the first question that they had they were concerned with is, what is an individual? When we as individuals emerge? And of course, there is a big variety of opinions, and we should be respectful of them. There is many people that think that you are an individual from the moment the egg gets fertilized, and I think that requires the same attention and the same respect that others. But this committee decided that the time at which the individual begins is the time at which gastrulation starts. And the reason for that is that is the moment. Before that moment, you can take a blastocyst, separate it into two, and you will get two individuals, two people. In fact, that's probably one of the ways that you can get twins. I mean, there are many, but that is one of the ways. And remember that individual means undivisible. So it is thought that the individual is the moment in which you cannot not divide it into two, and that's exactly what happens in gastrulation. You can get Siamese twins, but that's a totally different story. They are just one people that has two bodies. And they decided, therefore, that you could not, if that is the moment at which the individual arises, which is the beginning of gastrulation, the beginning of gastrulation was the time at which, if you are culturing embryos in vitro, you should stop and you not continue that. That's what it's called the day 14 rule, okay? Now, this was in the 1980s, and of course, a lot of rain has fallen in England, and a lot of time has passed into this, and people are developing techniques, and over the last few years, some people have managed to grow human embryos in vitro until day 14. They claim that they expend their experiments at day 14 because of the Warnock report and the Warnock rule. I don't think those embryos are very healthy. I don't think we have a long way to go to actually get gastrulation in vitro from human embryos, because the techniques are developing fast and people are curious. Over the last 12 months, there have been a lot of encouragement to revise this rule and allow the development of human embryos in vitro beyond day 14. I think it's going to be difficult, but there is a lot of talk of revising the day 14 rule. But in the meantime, we would like to learn about gastrulation. I have shown you an alternative system that allows us to study gastrulation in the mouse, which is based on ESLs. And so the question is, we just wondered in the lab whether we could take the techniques that we've learned from using embryonic stem cells in the mouse and transplant them to the humans. And many people have done that in the beginning for the pre-gastrulation stages. These are some examples. But the question is whether we could build gastruloids out of human embryonic stem cells or induced pluripotent stem cells. And indeed we can. And again, I want to emphasize this issue of reproducibility that is very important to us. We are not interested in events that happen once in a blue moon, but events that we can reproduce. And basically the results that we have obtained with human embryonic stem cells and human gastruloids are very similar to the ones that we got in the embryos. You can see that when we apply the stomosig, we see an organization of genes along the anteroposterior axis of the gastruloid. And when we compare this to the mouse embryo, because that's all we can do in a high throughput manner, we find some interesting differences that, of course, we don't know if they are due to the fact that these are gastruloids or to the fact that there are differences in the human and the mouse. But I think this is a first step towards that. We also get representatives of the 3-gem layers, and when we look over time, we can see that it's very reproducible. This is 3 samples, so this is RNA-seq of different samples over time, and we get a cadence, a sequence of gene expression that tells us that these cells, as they are forming this structure that we call a gastruloid, are going through the motions of gastrulation, because the genes that we see expressed in time, the program that is unleashed by this is very, very similar to the program that is in mouse gastrulation or in other vertebrates. We also have expression of the Hox genes in an order, and I don't have time to show you, but also spatially that tells us that these things are really recapitulating many of the things that are typical of at least a mammalian embryo. We have a heart primordium at the end, and when we look, we can see a great deal of the genes that are associated with the primordium for the heart. We haven't got to a stage yet where these becomes cardiomyocytes and they beat, but they are there. The potential to develop that is there. At this structure, at this moment, as you can see, the heart is the most anterior part of the gastruloid. And you might be very surprised to hear that. Of course, they don't have a brain, but then turns out that in the early stages of development, because of the way gastrulation happens, this is true of the mouse and of human embryos, the most anterior part of the embryo is the heart primordium, which is pushed back by the growth of the brain. But actually, along the anteroposterior axis, the most anterior part during the 3rd week of development in a human and the beginning of the 4th week is the heart. You can see here as the brain is starting to go, the heart is being pushed back. This is from some of the sections of the Carnegie collection. Now, to see whether this is recapitulating some of the events that happened in the embryo, we treated the gastruloids with an inhibitor of nodal. Nodal, it's a signal that it promotes endoderm. If you don't have endoderm, you're going to miss the heart because those interactions are very important. And basically what I'm telling you here is that when you apply SV43, which is an inhibitor of one of the SMATs, the gastruloid lacks a lot of the endoderm, and as a consequence, you do lack the cardiac. Now, what do these structures have to do with the embryo? Well, we were, again, not surprised at this stage to see a signal, very strong signal of somatogenesis. That's what it is. And now we can see that if we go to the Carnegie collection, we can see that at day 17 to 19 of development, you don't have any somites formed. But at day 19 to 21 days, somites are forming. Because we have genes that are expressing somites, by the argument that I told you before, this means that the gastruloids are related to a time that the embryo has started to make somites. And we reckon that our gastruloids are related to embryos of about 19, 20 days of development. Still, the issue of the, so therefore we have a system that it may allow us to understand human development, but it would be very good to have a validation. This is very important, and this is one of the missions that I've set up myself over the next years to develop, to try to bring the Carnegie collection up to the 21st century and trying to collect data that will allow us to compare these embryo models that we are using with real embryos. In the meantime, just accidentally in the UK, the group of Shankar Srinivas got hold of an embryo, a human embryo during gastrulation. they had to make a decision, and I think I really commend them for having done it so well. They decided to do RNA-seq, single-cell RNA-seq, with this absolutely remarkable specimen. When they did that, the work has been published, and they managed, they don't have spatial information with the RNA-seq, but they decided to compare their RNA-seq with genome information from the gastruloids, and they found that exactly the same cell types that we found in the gastruloids, they are in the embryo, and they use the positional information that we can get from the gastruloids to organize the data. So this hints at a good validation, but is not total validation yet. So in order to bridge, while we wait to see if we can obtain material from human development to analyze our gastruloids and validate them, for which I will show you in a minute, could be good models for disease. In the meantime, we started a collaboration with Ramiro Alberio in the University of Nottingham in the UK. Ramiro is a farmyard biologist, and he's been developing embryonic stem cells of different farmyard animals. The pig is a very interesting one. Some of you might know that there is a lot of work for transplantation based on the pig. But one of the things that he decided that if he could do pig gastruloids, we could be able to compare the maps on the gastruloids to the embryos, because of course there is no ethical problems obtaining pig embryos, and here you have one. I'm not going to go into the details of the differences between the embryos, because even between embryos that have a flash disk epithelium or epiblast, there are differences, but it's a useful proxy for human development. But you can see here very beautifully, these are their pig gastruloids, which resemble a lot the mouse gastruloids. I'll be happy to answer questions about this. That would go into a totally different topic of the similarity of the different gastruloids from different species. What we've been able to do is to compare, again, RNA-seq from the pig, from the mouse, from the human, and we find that the pig is, that's all you need to know there. It's a very good model, we think, for human development. It's going to allow us to build a bridge between the gastruloids and the human embryo, which would be very difficult to build from the point of view of the mouse. So, as I say there, we have a system. We think it's early days. It's a prototype to study fundamental biology, properties of living matter, compare the human and the mouse, which is important to see what lessons we can take to the mouse and which lessons to take from the humans. We can learn about gene regulatory networks and epigenetics. We can also study timing of developmental events. The early development in the human is lower than the early development in the mouse. We have here a system, and the gastruloids seem to be recapitulating that, so this provides a system to study that, particularly because of its reproducibility. We can create integrated organ systems like the heart one day, or niches, because they require interactions between different groups of cells that organoids, because of being directed in particular directions, they are missing. But here we are recreating the primordia of the different organs and tissues interacting in a very confined space. And also we can use them to disease modeling, and this we started in a collaboration with Chantas Aleph in the Ashby Institute in Kyoto. And I told you that these gastruloids, they are the beginning of somatogenesis. Chantas Lab, in collaboration with Naomi Morris, now in London, in the Creek, they managed to push the system such as that you can see here, they are getting somatogenesis happening in human gastruloids. These are from iPS cells. Again, much as in the case of the mouse, we need to include them in Matrigel. Something that I didn't mention, but might be known to some of you, is that the rhythmic generation of these somites is associated with periodic gene expression of a number of genes which run through the posterior to the anterior part of the body in waves. And turns out that the gastruloids recapitulate that. This is a movie of one of those genes. You can see that as the gastruloid is extending, very much as it would happen in the embryo, you are getting these waves of gene expression. Then when we reach a front, when they reach the front, they generate a somite, and they start again, and they start again. So this is also going to give us an opportunity to study these processes, and they do it with the period of the human. So what Chantas and Naomi are interested in, as indicated in the next slide, is in whether we can use this system to study spinal cord defects and cage defects. These are some of the diseases that have been associated with spinal cord defects, and these diseases have been mapped to genes that are known to be involved either in the periodic expression of these genes or in the generation of the somites. And what basically we are showing you here is that we can put these defects or use iPS cells from patients with these defects to generate gastruloids that are making somatogenesis, and now we can learn where is the origin of some of these systems, and I think it's going to be useful. Something that this could be used for other diseases, and we hope that will be the case. Something that I never thought I would be interested in, or this would be useful for, is as a system to studying early teratogenicity and toxicology. And this is work that we did with Roche, in which we basically, this was a pilot study in which we wondered whether we could use the gastruloid system to study the effects of several drugs on the early stages of development and gastrulation. It's been published, and I just to show you that it's promising. That's what I will do. I think there are many. I learned that there is a lot of issues in this field of teratogenicity, but you have to realize that this field uses mostly animal systems, and that here we are providing something much more directly related to humans that bypasses the animals, and that can be very useful for these studies, and we are hopeful that this will be the case. Now, I have to come, so there is hope as long as we remain with the baseline low, as long as we acknowledge the difficulties and the limitations, and this is why I do have problems with these statements that maybe have to do with our attempt to play God or to saturate our egos or our interest. And that gets worse because people then, things that have happened in the mouse, they can get extrapolated into humans without anything, and then you start alarming people, and that can feedback on the research. I think this is why we have to be careful with the way we hype the research to the journalists, because when they find out that we haven't done what we said we have done, that will have a very big backlash on what we do. And just to tell you, remember that I told you that these things, these structures that have raised, and I think the scientists doing this are guilty of that, have generated all these tears, happen at 0.5% of the initial thing. And they are incomplete. They basically 0.5% abortions, aborted mice, abnormally aborted mice in different ways. Let's look a little bit of history. Turns out that Louise Brown, the first in vitro fertilization baby, is the result of 282 implantations that led to 5 pregnancies and 1 baby, but that baby was perfect. And it went to term. And that's why in vitro fertilization is what it is and why it was noteworthy. Dolly resulted from 277 clones that were implanted. That led to 29 embryos and 1 sheep. But that sheep was perfect again. So it's newsworthy. The Yamanaka protocol, which is the induced pluripotent stem cells, is usually 1 in a million or 10 million cells, but it's perfect. So I do object to the point that we cannot be playing with things, that we cannot say that we've reached a goal that we haven't reached with these numbers. These things are useless, and I'm very glad that some people, this is one of the 2 authors, this is Jacob Hanna, that always he makes the point, synthetic embryo breakthrough, yes, but growing human organs is very, very far off. What these models are how, this is a very famous line from Victor Hamburger, a real teacher has been and still is the embryo, who is incidentally the only teacher who is always right. But I hope I have convinced you today that when you play with cells, when you ask cells to do things, you discover things about embryos, about how we are made, about how they made themselves, that we could not figure out in any other way. If you think of a Spemann experiment, that taught us a great deal, not by reconstructing an embryo, but by playing with the cells and putting them around. Drisch experiment, which is one of the separating cells, you learn things that you don't expect from embryos. The genetic code, we learn the genetic code by taking the system apart and taking it in vitro. I think we are learning things about embryos that genetics hasn't taught us, and it's teaching us something about cells that we still don't understand. But if we do that, we are going to be able to build better organs and systems in the future. So thanking you for our attention. I told you that it wasn't going to be provocative, but there were 2 parts, the part 1 and the part 2. I just want to thank, this is the group in Barcelona that we are working on some of the questions that are with us. This is our collaborators. Naomi Moore is now in the CRIC, was very, very important, particularly in the development of the human system. Chantas is now collaborating with her in developing the SOMITE system, which is already in the archive. And particularly, as I said, my collaboration with Denis Duboul and Matthias Lutolf in EPFL was absolutely essential in launching the system. I'll be happy to answer questions now or at coffee, and I thank you for your attention.